Well, welcome everyone to the weekly Forever Green lab meetings. And just remember the intent of these lab meetings is to get an update on the various Forever Green projects or associated projects and then take the time to necessary for discussion and thinking about future partnerships and future endeavors. So I thought it would be a great opportunity. Deborah has given us an update on the alfalfa protein work over the last number of years, but she now has, I think, and I've asked her to take the opportunity to introduce the redeveloped USDA plant science team. And a lot of you know that over the years, this plant science team played a key role in the development of alfalfa in the United States. And this team, as a result of retirements, dwindled down for a period of time, but now it's back up at full strength. And I wanted to make sure that everyone was aware of that, right? All of you that are associated with the Forever Green Initiative, the alfalfa program, forage program, the legume program is back in full force, and looking forward to Debbie Semick, the lead scientist for the USDA plant science program, to give us an introduction to her team members and to give us some ideas of what the program may be thinking of and focusing on over the next number of years. But historically, this has been a major part of our plant science program on the St. Paul campus, and we're really excited to have it back in full force again. So with that, Debbie, I'll turn it over to you to introduce your team and give us an update.
Great, thanks very much, Don. And I apologize again that for some reason, my video camera is not cooperating, is not working today. So you'll just have to look at my little thumbnail picture. So I am really happy to have this opportunity to give you a little bit of a research update and to introduce the complete ARS Alfalfa project team. The research update I'll be giving you is from a NIFA funded project, which is in collaboration with Dongfeng Den and Brian Shepard and Matt Digman. And with that, I will tell you a little bit about what I'm going to talk about. So there's actually going to be three of us talking. I will give a little bit of background on the Alfalfa project, introduce the current members and our research goals, and then give you that research update on what I'm doing in Forever Green related research. Then I'll turn it over to Zhenyu Xu, who will talk about his alfalfa breeding program, and Josh Gamble will talk about his agroecology soil science program. So a little bit about the Plant Science Research Unit. We are actually 65 years old. So this is our Blue Sapphire Jubilee for the Queen of the Forages. The first hire was a plant pathologist in 1956. And this Fred Froschheiser was hired to work with a University of Minnesota breeder, alfalfa breeder. And Don Barnes actually joined that project in 1965. What they did was to do a lot of disease surveys. They developed selection methods for different diseases, and many of those methods are still in use today. And then they developed some of the very first disease-resistant germplasm to major alfalfa diseases, notably cultivar called Agate which was the first alfalfa cultivar with resistance to Phytophthora root rot. And Don and Fred had a very productive career together. They developed many of the methods for breeding alfalfa and developing specific traits into alfalfa, particularly for disease resistance. And they ran disease nurseries for the industry for decades. As a program developed, they moved into, Joanne Lamb especially worked on forage quality and developing different root architectures in alfalfa. There's also a very strong plant physiology component with Carol Vance, as particularly looking at nitrogen fixation, as well as Gary Heichel was part of that group. And with Craig Schaefer, they released the very first legume crop that had been bred specifically for nitrogen fixation capacity. And this variety is called Nitro. And it was released for use as a cover crop and as a green manure crop. And it is still on the market today, although it doesn't get a whole lot of press. And so, I would encourage folks who are working on cover crops to, put Nitro into some of their trials as comparison. So it is a fast growing kind of annual summer alfalfa, and it was bred for high forage and biomass yield. It has deep taproot, so it can help with compacted soil and sort of mobilizing nutrients. As the program matured, a part of that nitrogen fixation work was the discovery of alfalfa mutants that actually are incapable of fixing nitrogen. So they're ineffective. And these were used very effectively in a lot of bioremediation trials. And this picture up on the right corner is Michael Russell and Joanne Lamb at the site of a train derailment in North Dakota, and where the ineffective alfalfa is being used to clean up this nitrogen contamination in the soil and groundwater. And the ineffective alfalfa, because it can't fix nitrogen, it's just taking up nitrogen, it turns yellow when the nitrogen levels have been decreased in that field. Michael did quite a bit of work on the environmental services of alfalfa, measuring the nitrogen credits from alfalfa production, as well as nitrogen capture from surface and groundwater. And then there was quite a bit of work on developing alfalfa as a bioenergy crop and developing a high biomass non-lodging alfalfa that you can see here. And one of the advantages of this non-lodging alfalfa is that it can be harvested just about twice a year because it doesn't lodge. You can still gather that herbage. And it can also flower, so it can be very effective bee pastures. So for pollinating insects to get nectar and pollen. So in my group, we worked with Carol Vance in biotechnology, developing very efficient alfalfa transformation methods and introducing novel traits into alfalfa, as well as using transgenic alfalfa to study gene function. With Carol, we developed the very first transcribed gene atlas for alfalfa and worked extensively in Medicago truncatula as a model system. And as Don mentioned, over time, we lost 6 of the scientists to retirement from the group. And for a couple years there, it was pretty grim because the alfalfa project was basically just me and a bunch of technicians. But we have a really strong stakeholder group in the National Alfalfa and Forage Alliance, NAFA. And this is one of their graphics showing that alfalfa is America's 3rd most widely grown crop. In about half of the states, it is among the top 3 of the field crops. And you can see it's number 1 in California, as well as a number of other states, with the highest producing states, California, Wisconsin, and Idaho. So they were very successful in getting us new funding to be able to rejuvenate the alfalfa project. And we are now back up to full strength. We have 6 scientists working on alfalfa with plant pathology, Josh Gamble in agroecology, Jan Yu in genetics, Joe in forage biochemistry, Dan in soil microbiology, and Sean in legume genomics and gene editing. Now, the alfalfa industry really faces very difficult problems in that there's been very dramatic decline in alfalfa acres over time. And this has really been because more land is shifted toward production of row crops, as well as because our animal numbers, especially dairy, have really declined in the Midwest. And corn silage has basically replaced a lot of alfalfa in dairy cow diets, mostly because of its high yield, very uniform quality, and reduced labor for producing this feed. So how do we reverse the trend of declining alfalfa acres? Well, when we talk to producers, it's really these four different trends. So we need to increase yield. We need to decrease the labor involved in producing alfalfa. We need to decrease the economic risk of producing it, which includes risk of winter kill and winter injury. And it would also help to increase the value of alfalfa. We need to demonstrate its economic value on the farm. With the new leadership in Washington, we'll probably see some payments for carbon credits. And so alfalfa is excellent at storing carbon, and we need to demonstrate and value that, as well as developing some new uses. So alfalfa is probably the only crop that you can think of where it's basically just fed to animals. We don't refine it into multiple products. A few people have developed some other sort of alternative products for alfalfa, and I just recently saw this in my horticulture magazine, which is an advertisement for what's called Alfalfa Secret. And this is a producer in Minnesota who is marketing this to a lot of the green industries in the state for using it as a fertilizer as well as a mulching component. And then one of the products that we've really been interested in is concentrating the protein from the herbage into a high protein component that can be used for animal feed. And so, and which I'll be talking about in a little bit later here. So as the new unit has been redeveloped, our overall goal is to increase the footprint of perennial forages on the landscape. And we're developing alfalfa for livestock utilization, environmental protection, and soil health. And so our different activities are to increase yield, improve forage nutritive value, improve stress resistance, enhance the environmental services of alfalfa, help to develop novel uses, as well as develop new breeding methodologies. So overall, the goals are to have cooperative projects within the unit, within ARS, and with our university industry partners. We want to develop tools in germplasm that will be useful for growers, for extension educators, and commercial breeders. And eventually, we really want to cover more ground with perennial forages and increase alfalfa acreage. So I'll just quickly go into some of the projects that we're working on in the area of developing genomic tools. When we wrote our project plan, believe it or not, there was no reference genome sequence for alfalfa. And part of this is because it is an outcrossing autotetraploid, which makes genome sequencing complicated and quite a bit of a challenge. Chinese group has since published 2 different reference genome sequences. They're pretty high quality. However, they are using Chinese germplasm. And we have in in progress now development of a reference alfalfa genome sequence using US germplasm. We hope to have a really gold standard alfalfa genome sequence that identifies all 4 haplotypes in the genome, and then to develop draft genome sequences of at least 10 different alfalfa plants that vary in their fall dormancy. Then Sean Curtin is developing more efficient ways to do gene editing in alfalfa, which again has its own challenges because of the autotetraploid nature of this crop. We also really lacked just some of the basic tools for breeding, molecular breeding in alfalfa. We didn't have universal SNP markers or a SNP chip or anything like that. And ARS has put quite a bit of funding into a group called Breeding Insight, which is out of Cornell University, to help us develop universal SNP markers in alfalfa and have very economic platform for doing genotyping in alfalfa. So with those markers and with a reference genome, then we'll be set to start doing marker-assisted selection, genomic selection in alfalfa. And we'll be working on mapping a number of genes of economic interest, agronomic interest. We have material ready to do marker mapping of disease resistance traits, forage quality traits, and root architecture traits. And with Zanju's skills in bioinformatics, he is also bringing in artificial intelligence and machine learning to alfalfa breeding. He'll be talking to you about. So some of the breeding goals are, again, to improve all of these things: yield, winter survival, seedling disease resistance. And here I have a picture of some of the problems we had last spring. This was a heavy wet soil over in Wisconsin where we have a very poor alfalfa seedling establishment, a lot of disease and root rot going on here, compared to a soil where this is what you'd like to see, very strong, robust alfalfa seedling establishment. We're working on increasing fiber digestibility. This is for ruminant animals, and also increasing the amount of protein in alfalfa for some of the alternative uses that we're interested in. And because of the increasing wet falls in particular that are impacting winter survival, we're also looking at breeding for improving tolerance to wet soil. And then we have a number of sort of proof of concept studies going on to increase energy and reduce methane production with a transgenic approach. And then through gene editing, Sean is working on developing alfalfa for phosphate accumulation, as well as increasing seed size. So Josh will talk a little bit about increasing alfalfa utilization for environmental services, some of the novel forage rotations that he's investigating, doing this process modeling in dairy agroecosystems, and also the effect of dairy manure on crop yields and on rotation. Then I'll talk just a little bit about this alfalfa protein concentrate and how we have investigated it in use in aquaculture feeds. So I pulled this graphic off the web. I wasn't aware of this group doing this research previously. This is a company called Transparency Market Research. And they are looking at the global alfalfa protein concentrate market share. And they are predicting that there's going to be about a 7% annual growth rate in this feed ingredient. And they're looking at it as in dietary supplements, animal feeds, and pharmaceuticals. And so they're looking at, this is, $375 million global market in 9 years. So I'm not sure if this dietary supplement, if they're talking about human food uses or as just as a supplement, say in smoothies. But of course, we're still trying to get interest into using alfalfa protein in human food products. We thought that aquaculture feeds were a really good place to put alfalfa protein because it is a very uniform kind of protein. It shouldn't have a lot of anti-nutritional ingredients in it. And that in aquaculture feeds are really looking for new proteins to replace fish meal in formulated feeds. So we got a grant through Minnesota Department of Agriculture, and Jessica Coburn was a master's degree student in the APS program who worked on this for her thesis research, looking at the yield and composition of this protein concentrate, and then did feeding studies with yellow perch and rainbow trout. And so we used this non-lodging alfalfa that we either just harvested the total herbage or we stripped the leaves and made protein concentrate just from the leaves. So we juiced the alfalfa, and then from that juice, we can precipitate the protein using different methods. And then it was also incorporated into these fish feeds. So the results of her project that I presented this last year was that with the different ways that she was producing the protein concentrate, either with heating or with acid or with freezing, we could modify the composition of the final concentrate. It was all about 50% protein, but then the remainder could vary quite a bit in the amount of sugars and lipids that were incorporated into the concentrate. We also found that the amount of protein varied over the season, over the different harvests. We got the herbage had different amounts of protein in the leaves. So this really opens the door for doing research to look at management as well as different varieties of alfalfa to optimize the amount of protein produced. We were really kind of surprised with the amount of protein concentrate that we could get out of the biomass alfalfa. So we had an early spring harvest that we got about 4.5 metric tons of dry hay. And then over the next 3 harvests, we accumulated 854 kilograms of the protein concentrate, which again is about 50% protein. We also got about 7 tons of dry press residue, which has most of the protein removed, but not all, and could also be used as an animal feed, primarily for beef cows or for dry dairy cows. But what was really surprising was that we got 42% more of the protein concentrate using the biomass type alfalfa than using typical hay type alfalfa that was harvested at immature stages. So for the companies that are developing or producing alfalfa protein concentrate, might pay for them to actually switch to this biomass type alfalfa. The other thing that Jessica found was that the protein concentrate, replacing this in perch diets, replacing the fish meal, was very successful, and that the perch accepted the feed, but we did see some delay in growth. So with this sort of preliminary research on the palatability of the feeds containing alfalfa protein concentrate, we were able to get funding through NIFA and collaborating with aquaculture specialists at the University of Wisconsin in Milwaukee and within ARS. And then Matt Digman is conducting an economic analysis of the production of this protein concentrate from field to the concentrate itself. So I'll only talk about a couple of objectives and the work that's been done so far. The first part was to really take a better look at the feeds that have the protein concentrate, so that to answer a lot of the questions that some of the commercial producers had. They wanted to know, how does this affect the physical quality of the feed pellets? What about palatability and digestibility? And we're using rainbow trout. And with this, we should be able to establish an optimal amount of the protein concentrate in trout feed and carry out a commercial feeding trial. And so here are some of the setups at the University of Wisconsin-Milwaukee. So here are some of their results. First, looking at the durability of feed pellets that are produced with different amounts of alfalfa protein in the diet. And so this is the durability, means how well the pellets stay up with different handling and manipulation, so that they're not falling apart in shipping. And you can see that we did see a little bit increase in durability from the feed that was made with just fish meal. So this is a good thing, that the pellets retain their shape during shipping. Then when they're put into the water for feeding fish, increasing the amount of alfalfa also increase the stability in water so that they aren't falling apart as quickly. And so being more available for fish for feeding. And then they also tested the density of the pellets, the feed pellets, with increasing amounts of alfalfa in the diet. And they did see that the pellets become more dense, or sort of harder in their typical way of making these pellets. So this was a little bit of concern in that it might affect how well the fish accept these pellets. So when they put them into digestibility study, this was a very nice thing to see, in that with increasing amounts of alfalfa in the diet, the digestibility was just as good as the control diet, the fish meal containing diet. They also saw no significant difference in feed intake with increasing amounts of alfalfa in the diet. So another really good result occurring from this. The only downside was that the amount of phosphorus that could be extracted from the feed significantly decreased with increasing the amount of alfalfa in the diet. So this really is of concern and needs to be addressed. And luckily, part of the grant was actually to test how well we could liberate phosphorus from the feed. This was part of our second objective, was to look at the effect of an enzyme called phytase when we incorporate that into the feed. And so we know that a lot of animals have trouble getting the phosphorus out of plant feeds, because the phosphorus is often tied up in it's called phytate. So it's all bound to this inositol molecule. And the enzyme phytase then liberates the phosphorus, making it available for animals, as well as liberating a lot of the associated mineral nutrients. So one of our objectives is to use commercial phytase, which is often used in different animal feeds, to add this to the trout feed to see how this impacts digestibility of the feed and utilization of the phosphorus. So they'll be measuring growth and health of the fish, as well as water quality, because we don't want that phosphorus to be leaving the system and contaminating water supplies. We also have a transgenic alfalfa that is expressing phytase. And so we're putting these out in the field this summer so that we can get an idea of how much is being produced under field conditions. And we'll be quantifying the amount of phytase produced in the plants. And then once we juice these plants to produce the concentrate, how much is in the juice, how stable is it, and how much is actually in the protein concentrate. And is it still enzymatically active once it's in that protein concentrate? So we'll be testing the different methods of making the concentrate like that Jessica developed to see if we can vary the amount of phytase that's in that concentrate. And that is the end of my part. And so at this point, I will turn it over to Zanyu to talk about his alfalfa breeding program. And I think we decided that I would just advance the slides. So he can just tell me when he's ready for a new slide. So thanks everybody for listening to me.
So Dan, you want to the question, the Q&A for each individual first, or you want to wait at the end of the presentation?
Let's hold them to the end.
Okay. So my presentation is about alfalfa breeding. I want to use a powerhouse maybe to emphasize our team. To my knowledge, this center or this unit is the best alfalfa breeding center. And as you know, as you may know, some of you, I coming from a commercial breeder company. I always ask, why majority of the cultivars coming from industry, very few coming from public. And there may be more than one reason, but one of the reasons is a breeder can get fully support from a company. They have upstreams for the marker discovery, gene discovery, and downstream for the agronomists, etc. They really do not have many breeders. For example, Syngenta, the second largest seed company right now, they only have like 6 or 7 soybean breeders, but their market like 15% of the whole world market. Harris Stein instead, they only have 1 soybean breeder for their whole company. And that breeder, or maybe one interesting is that breeder, the richest breeder in the whole world. He has his own jet plane, travel for business, etc. Oh, a lot of breeders, including me, want to be that rich, but definitely probably in my lifetime cannot reach there, except a lucky lottery someday. But the main points I want to point here is a successful breeder is coming from a collaboration, coming from a teamwork. And so what I show here, as a breeder, new breeder, I will start my breeding program. Debbie has hired the new SOIs, the new scientists. I treat those as my upstream supporters or scientists. You can see here, Joe is the biochemist, and I collaborate with her or working together with her to breed for digestible biomass or alternative usage of components. And she and I talking about what special trait that we can have another products that farmers like and benefit for human. So from Dan, he's a microbiologist, what kind of microbes that can increase both the yield and also for the environment. And Sean, Debbie mentioned several times, he's expert for the gene editing. He already developed the phosphor uptake, the 2 lines, and we will make a crosses this year. And Debbie definitely is the leading person here for the biotechnology, and the markers, the sequence, etc. That will help my breeding program using genomic selection, machine learning, etc. Josh is a cropping system guy, and I work closely for any cultivar specifically good for cropping rotation or cropping combination system. So I'm the guy is put all your discoveries and science into the farmer's field. So again, my point is a breeder success coming from upstream and downstream support. So next slide, please. So with that structure, with that strong support from the team, where and what I can do in the next 3 or 5 years. So I'm thinking about the following 4 parts. Number 1 is gene editing, the most promised technology for the breeding. I Sean already have 2 phosphor uptake, and we plan to make 14 crosses. And also the other thing is I have been working for hydrolysis for many years for soybean and alfalfa. So I'm still passionate using the hydrolysis to improve the yield. So there's a new technology, you use gene editing, you can edit any line becoming sterile line, and any line, the wild type of that line is a restorer line. You can make a crosses without the traditional 3 line to use the hydrolysis. And my master degree for the hydrolysis, I made 40 crosses. The highest is like 45% hydrolysis. With male sterile line, my supervisor was developed. And I will continue this study or continue that trend using new technology with the gene editing. And the other thing is for the self-inbred depression and self-incompatibility, if we can overcome that and use the hydrolysis, that probably will break the yield bottleneck for the yield improvement. And the second part is for the artificial intelligence with the machine learning and deep learning. I have a slide basically introduce the stress cam and the drone, etc. And genomic selection right now is applied to almost everywhere. And the last part is the traditional breeding. So no matter what kind of technology you use, it's basically to help you improve the breeding genetic gain. You use marker assisted selection, make a precise selection or accurate selection. You use UAV to improve the phenotyping, make sure when you do marker association, you make sure you find the marker is correct. When you use machine learning or deep learning, when you use the algorithm to learn from the phenotypic and genotypic data, you make sure your phenotype data is reliable and repeatable. So all those depends on all those technologies increase the traditional breeding, you make crosses, increase the genetic. So that is the four areas I will work on. So the coming slides, I will go over 4 aspects. Next, please. So number 1, so is AI, artificial intelligence breeding from live phenotyping. So I want to emphasize live phenotyping. In the past or still current, the phenotyping basically is 1 time point or 1 data points. Doesn't matter the breeders go to field, well, you use a drone to fly in the field. You go there by a limited time or limited day when weather conditions allow. So what we propose here, and also this is the first time using this technology for the perennials. This technology has been applied for the soybean breeding for the annual crop. The first time for perennials, what we try to do is the stress cam with the power, solar power, with the Wi-Fi, they take image anytime any day, so year round. And then you can check your phenotype from your computer, from your tablet, etc. And you do not need to consider whether it's a raining day or cold day, you don't want to go to the field or too hot in the summer. So basically the system can take image anytime, then they can make a prediction from the machine learning model or deep learning model. What I show here is a stress cam in the soybean field, and also we installed in the alfalfa field, but we don't have a nice picture like this one. We have a picture to show in the snow, the later fall install. And then the USD has a collaboration with Microsoft. Microsoft has the artificial intelligence platform called FarmBees. So they have a strong team. They are put a lot of investment on the farm production, farm industry. So they developed the FarmBees, especially using the image analysis to predict the phenotype or production, etc. And USDA also built an artificial intelligence excellence center. They collaborate with Microsoft FarmBees and do the machine learning or deep learning, etc. Then here I show a picture with alfalfa covered by snow to show you our stress cam can take image anytime. So you don't need to care about the weather is too cold. You can take an image anytime. And eventually what this can help us is you do not need to do any destructive sampling, or you go to the field, you take many data points for the phenotype, and you can do any genomic level work, genomic prediction or selection, etc. This is for breeder. And for the farm, you can use this technology to diagnose water usage, fertilizer application, etc. And the stress cam, this system is only, the stress camera itself is only $150 a piece. But for our research, since we including all the solar panel, etc., is more, it's probably $500 a piece. I spend more time for this slide because this is used for every phenotype and that can be used for almost every aspect of my breeding. So next slide. And as Debbie mentioned, the protein, this is where my passion is. So alfalfa on the top left side, you can see the clear contrast, how much more alfalfa can produce protein, and from each acre. This is the average only, and the top right side is show what is the potential, how much more alfalfa protein we can get per acre. What I show here is the previous work. They extract the crude protein, NDF, ADF, etc., from 1,256 genotypes. You can see the percentage of crude protein, over 30%, they have quite few from the leaf, from the stem. And I have requested the top 200 PIs for my breeding program to target further increase of protein production. And the other side, on the bottom right-hand side, you see the protein degradability for the dairy cow, a high percentage, the bottom is the percentage, protein will be degraded before the, in the rumen, before they digest into the instant for amino acid or minerals, etc. So we needed to breeding for high bypass protein. So my goal for breeding will breeding high total protein, a high bypass protein, a special protein for food protein. And I use the term, I borrowed the term from Craig Schaefer as alpha burger. someday if whether someday we come back from the restaurant. So next slide. These slides show the work, previous work from Joe Lambs. What I show you here to see how successful for the digestibility, this is the stem digestibility. On the left top hand is the low digestibility by low digestive population through cycle 0, cycle 2, and you can see if we cross low by low, your digestibility will go further. You see that clear trend. And if you cross our digestibility, high digestibility by high digestibility, you can see the digestibility in the past 3 cycle selections clearly increase. I put a line here to show they're on the same level at the beginning. Then you get low by low, high by high. You may ask the question, why are we breeding for low digestibility? So the ones for contrast, when we do marker association, we can clearly see where the positive allele, where the negative allele for next breeding selection. And the bottom right hand is the genetic gain for the digestibility, 16-hour or 96-hour digestibility. You can see clearly the genetic gain is significant. There are squares really high, and the p-value is definitely smaller than the threshold 0.01. So this is the digestibility. And next place is similar for the root architecture. And this one, the work I'm also excited about is we can develop a specific root architecture for the environment benefits. So what I show here is basically we have 2 different root architecture, taproot or branch root. Also on the right hand side, they show the genetic gain. And even though the genetic gain not as good, not as high as digestibility, but we make a progress. And with the universal marker Debbie talked about, my expectation the genetic gain will be much higher than the phenotype selection only. So next. So this for the special traits breeding, and the number 1 here is the omega-3 breeding. I talked about this, and I extremely passionate about this, and I discussed this with Dan a couple of times. So as you may know, Monsanto developed omega-3 soybean. They call it a game changer for 10 years. And they spent $200 million for this product. The gene itself is well expressed in the soybean seeds and is really useful or beneficial for human. But the challenge is during the process of the oil and the nutrients components get damaged, and farmers cannot make money from it. So Monsanto gave it up. Right now, I talked with Bayer a couple of times. They agree to share the gene constructs with us to do the testing, whether we can transfer omega-3 in alfalfa. And I do not know whether it's successful or not, but I do know like soybean, alfalfa is the legumes, they share similar genomes, and my expectations is probably will have higher probability to be successful, to succeed. And if it succeeded, then the potential will, to me, it will be huge. For example, you can develop alfalfa feed for fish or shrimp, or dairy cow, etc. You can have omega-3 fish from lake or river instead of the deep sea only. So right now, I'm request 4 gene constructs for the omega-3. I hope I get it soon and start the work. Number 2, Debbie mentioned in her lab, he has a high lipid. and I want to use that to breeding high high energy or high lipid alfalfa. And the third one is the phosphouptake, and this is through gene editing. And how good this material. And I know Sean is here. If I misspoke anything, please jump in. So the two line or the two material, they either have 7-fold high phosphor uptake capacity, and the other one is 4-fold high phosphor uptake capacity. They're really good gene editing materials. I'm very excited about this, and Sean and I designed 14 crosses for this year. We want to test this gene editing phosphor uptake, whether we can quickly transfer to commercial varieties, or how well this perform to the benefit of environment or to the benefit of the yield, etc. And the last one is the environmental trait. I'm a breeder. I present my breeding program to an environment protection meeting. From the meeting, there are several department dean. They said, "Oh, I will build up a department specifically breeding for environmental traits. In the past, we always breeding for either human or animal to make money. We never seriously breeding for environment. So I'm planning to do that too. Especially under the forever green, we want to cover the ground year round. So I want to breeding extremely cold with high yield and a special canopy to cover the soil and carbon to sequestration to protect, to reduce the carbon dioxide concentration, etc. So next slide. Again, I joined this team last year. I'm very passionate, but I'm not sure what is next biggest pitch for my alfalfa breeding program. But I do know if I continue to the traditional breeding, only work for ruminant alfalfa, probably not going anywhere. I may have a biased opinion. So, but my passion again, I want to breed for human food alfalfa, aquaculture alfalfa, or environmental alfalfa, small animal alfalfa, etc. So I want to keep this slides, and right now, design my breeding program with the idea in my mind is, let's say this is 2030, 10 years later, when we go over these slides again, where what I developed, what achievements I did, whether my research have some products in farmers field. And that is my last slide, but that is, you can see where my passion are. So thank you, and Josh, your turn.
Great. Well, I don't know how late we want to go here, but
we're scheduled, as you guys know. We haven't laid it out for an hour and a half. Okay. But we usually don't go the full hour and a half, but certainly we want to hear, Josh, your presentation. So we still have, over 30 people online, so go ahead.
Yeah, well, I kept it pretty brief anyway. I kind of wanted to give maybe just a high level overview of what I've been working on so far. And go ahead and advance the slide, Debbie. So I wanted to just present this, what I consider my overarching strategy, and that's to develop and evaluate cropping systems, incorporating alfalfa and other perennial forages and perennial crops that are intended to improve water and soil quality, protect against erosion, and provide resiliency in response to climate change. And I think especially that last one is a charge that I've sort of been trying to meet head on here recently. So go ahead, slide, please. So one of the big projects I've got going on, and many of you will know this, this is a Forever Green funded project, and I presented on this last October, I believe. In terms of our progress is the Camelina and Craig Schiffer in terms of guiding us with alfalfa. And I won't read through everything. You can see kind of what we've outlaid here, but I'll just to think about this sort of integrating the cutting edge expertise that's been developed in the Forever Green program and deploying these novel cropping systems out on the landscape to look at yield and economic potential at the field scale, but also some of the ecosystem services. And so the model for this study is a paired field analysis where we're looking at a perennial forage and grain rotation, which is an alfalfa and kernza rotation, in contrast to a summer annual grain and oilseed rotation, or at least that's how we're conceiving of it at this point. We've gotten through a corn silage into a winter camelina winter cover thus far, and we'll be going into a short season soybean this summer. And so that system may evolve yet. But the goal of this is really looking at the field scale. What are the responses in terms of yield, carbon and water flux, energy exchange, and then Jess is doing some work looking at sort of field scale variability in soil health measures as part of this. And so we're really hoping this can be a valuable contribution to the Forever Green program going forward in terms of understanding more about the environmental impacts or the ecosystem services from some of the novel crops and systems that are being developed in Forever Green. Slide, please. Oh, am I frozen? Are you all hearing me still?
Yep, we can hear you. Yeah, I advanced the slide.
Okay, oh, there we go. I think my connection here. Can you all hear me okay?
Yes.
Okay, good. So the second project that's been a big piece of what I've been doing over the last couple of years is actually a collaboration that got started when I was a postdoc. And there's a group called the Dairy Agro-Ecosystem Working Group among ARS, and we've got multiple sites across the country. And this is kind of what we're calling like a common experiment that we have. And I'm collaborating with Greg Johnson and the LTAR network on this, which has been a great help for me. And the objective of this work is really thinking about in modern dairy production systems, many of these producers are importing feed. And as a result, they end up with more manure than they have land to apply it to. And so that was kind of the driving, or one of the driving forces to designing this project, is thinking about how do we utilize that manure outside of the dairy farm. What's the benefit of that manure? If we can ship it further away from the farm and maybe apply once every 5 or once every 10 years, what's the benefit? We know there's a legacy effect of manure, but what is it in terms of yield? But then also thinking about impacts on soil properties, and specifically here, soil microbial community structure and function. And so this project actually got started with Sarah Castle, who was a postdoc in our unit. And I need to update the slide. Of course, Dan Schlatter is now taking the lead on the microbial community aspects of this work. But it's a focus of really trying to understand what are the longer term impacts on soil properties and soil microbiology of the manure. But then also the rotation we're working in is a corn alfalfa rotation. So through our treatment design, we're also able to look at the impacts of alfalfa on first and second year corn in rotation, and impacts that might be having on soil health measures. And so that's a nice collaborative project that we've got across multiple sites in our Dairy Agroecosystem Working Group network. Slide, please. Okay, so this project, alfalfa life cycle analysis, this is led by Joel Tollickson out at the University of Minnesota at Morris. He's a life cycle analysis expert. You can see we've also got a number of other folks on the team: Jared Goplin, Brad Hines out at Morris, Bill Lazarus in Applied Econ here, Craig Schaefer and John Graber, John out at Dairy Forage Research. And this is a NIFA alfalfa systems grant that we're about halfway through. And so one of the things that we wanted to do with this project, obviously, I think most of you are familiar with the life cycle analysis concept. We're quantifying the impacts of alfalfa systems in dairy supply chains. For my part, the piece that I am really working on is getting better estimates of soil carbon change and how that contributes to the greenhouse gas footprint in life cycle analysis. So this is something that kind of historically in LCAs, there's a lot of hand waving around what happens with soil carbon. And so our goal was really to do some modeling to help inform, what the impacts of alfalfa in a rotation versus, a baseline system, increased alfalfa in rotation, or other systems that we might do beyond just corn silage. And I think this goes back to, what Debbie had talked about earlier in her introduction, is that we've seen a loss of perennial forage acres and increasing usage of corn silage as a primary forage source in dairy rations. And so that's sort of the baseline that we're comparing against. And we know there are a lot of environmental concerns associated with corn silage production. So really in this project, trying to demonstrate sort of through that whole life cycle process, what's the benefit of alfalfa in the system? And so, yeah, there's, Bill Lazarus be working on the economic component. Joel is leading the life cycle analysis modeling, and I'm working, we've got a grad student working on this. We're doing some geospatial analysis, looking at alfalfa cropping patterns and how those have shifted over time to help inform some of the scenarios we're developing to model in terms of, what are the sort of environmental impacts of a shift more towards silage corn and away from alfalfa. Slide, please. And the last sort of major effort that I'll talk about, I think, is something that I started as a postdoc working with Gary Feiereisen and John Baker in the Soil and Water Unit. And we've published a number of papers from this effort already, but this is an ongoing collaboration where we're really looking at, trade-offs in carbon and water and nutrient flows with dairy forage production. So there's a nice 10-year data set that we've been working with, and I'm moving that, now moving beyond just looking at the data to start thinking about some process-based modeling, soil carbon modeling in particular. So that kind of ties back into the last project to some degree as well. But just looking at what are the implications of a shift more towards corn silage, as well as a shift more towards like liquid dairy manure management. So one of the big papers, actually it's in press right now at Ag and Forest Meteorology. I'm pretty excited about this one, but we really demonstrate that this shift towards silage corn has pretty dramatic implications for the carbon balance of these fields. We're losing about 5 tons of carbon per hectare per year on silage corn cropped fields. And the conventional wisdom has been that the manure application makes up for this large carbon loss, which is primarily because of the large carbon export in a silage system, harvesting 90% of the above ground material. But what we've found is that in modern liquid dairy manure systems, there's just not enough carbon coming back out to the field to offset those export losses. And even in the case of intensively harvested alfalfa irrigated system this was, we find that we're losing a small amount of carbon. So the process modeling work going forward is really to look at what are the alternatives. We want to start thinking about alternative approaches to dairy forage and manure management in these systems as a way to retain more plant-derived carbon in our forage production fields. I see a question here, Peter, how does our research on carbon sequestration square with Randy Jackson's finding on perennial pasture sequestration? Which findings specifically, Peter, if you...
I might mischaracterize these findings, but in the broad strokes, I understand that there is limited sequestration potential from basically all current systems, and potentially a little bit more in perennial pasture, but it might not be a significant carbon sink is my understanding of his findings.
Yeah, so in terms of thinking about the actual potential for these soils to offset greenhouse gas emissions, right? I think in general, what we've been finding is that that's probably consistent with what we've found, I guess is what I should say. And I think broadly the literature has been, there's a lot, there are some varying opinions on what is the role of agricultural soil in climate mitigation, right? Can we offset substantial amounts of greenhouse gases, through carbon sequestration in soil? And it's a debate, but I think in general, the findings are suggesting that probably no, at least not in terms of a climate, an amount of emission offset that would be meaningful for the climate. But I think there's still a lot of value in thinking about the sustainability of our soils, right? And that's kind of the angle that I've taken a lot, thinking about soil health aspects and sort of reversing soil degradation processes that have occurred under certain systems. I think what we've found is somewhat consistent with that. Although I think there's still a lot that we don't know about, especially mechanisms in the deep soil, deep soil processes and the role of, say, perennial deep-rooted crops in long-term carbon cycles below like 30 to 60 centimeters. There's still a lot to learn there.
Josh, but I think what Peter's bringing up in the conversation that's going on is that the politics around carbon sequestration is getting ahead of the science.
Oh, sure.
Right.
Yeah. I mean, there's a lot.
Yeah. And your big boss in Washington is basically stepping forward and saying that that agriculture is the solution to, well, not the solution, but is part of the solution to the carbon greenhouse gas issue, right?
Yeah.
And here we sit and we all know what you just said, right? That in most cases, even in these perennial systems that are part of a rotation, we don't show that these systems are in fact sequestering carbon, right?
We don't, but I would say, I mean, there's a lot of uncertainty still. So if you look at the soil carbon literature, and this is one of the things we're addressing. And if you listen to my Forever Green talk in October, I probably bored you to death about methodologies for assessing carbon balance.
Yeah.
Traditional soil inventories are not very accurate. And so, in those methods, we, over time, there were a lot of studies that did show we could accrue soil carbon in some fields or with some rotations. In sort of the eddy flux type of studies where we're measuring gas exchange, we haven't shown that as much, but there aren't as many longer term studies with those approaches yet. And so I think there's still a lot of work to be done to reconcile different methodologies for how we're assessing carbon balance and doing it over longer terms. And then again, getting a better understanding of deep soil carbon dynamics as well, which I don't think we have a great handle on.
But the fear is, as you know, and I'm not poking at you at all. I'm poking at all of us in that you know, that the politics are going out and basically saying that these systems, with even just with cover crops, sequester carbon. As you said, there's a whole bunch of other benefits, but that carbon sequestration issue is pretty iffy, even in, you know, the perennial systems that we're all aware of. And Randy Jackson's long-term studies basically say the only system that has potential or has shown some potential to sequester carbon is our long-term rotational systems, right?
Yeah, no, I mean, I agree. The excitement around carbon credits for farmers and these carbon trading programs, I mean, they have outpaced, I think, the research to some degree. But, you know, just as an example, in a dairy system, if we're growing 90% silage corn and we're losing 5 tons a year, we can do better than that. We know that, and that might not be.
But I'm saying that's probably what we should be saying, though, that we can reduce that loss.
The framing of the discussion, could use some more scientific input, I think.
Right.
You're right. Yeah.
So, are there any additional questions?
Yeah. This is Carmen. Back in the late 70s and the early 1980s, I talked quite a bit with a Dr. Mark Honeyman from Iowa State, who was doing some real significant research on feeding alfalfa to swine. And I'm wondering if, looking at the development now of protein and separating out protein and making it available, has much work been done along the lines of introducing alfalfa into a swine ration, or is there anything along those ideas?
This is Debbie Samick. So when I brought this up to folks that work in the swine area, the issue has mostly been that we don't currently have production of enough alfalfa protein concentrate to go into commercial production, where they need basically, you know, train car loads of the material. So that's been the major stumbling block. There were, you know, small studies done that showed that, you know, it can be a good protein source for swine.
So the research data supports what Carmen is saying, that it could be used, right?
Right.
Okay.
Yeah, glad to hear.
Yeah, because I'm thinking if we could start substituting alfalfa for soybeans as a protein source, we look at carbon sequestration and we look at ecosystem services. I mean, there's no comparison.
So this is Jeff Berg. So shout out to alfalfa, says me, the former Wisconsin dairy farmer. So my question is probably in that vein, similar to Carmen as a farmer. So, and maybe related to the discussion about the ecosystem services. So with alfalfa, are we using that because that's the traditional forage crop? So we're ahead on the research versus, say, a clover. And then when we measure, how do we measure, success? So is it then we talked about digestibility, and I assume that means to a ruminant, but that may not be the same answer of measure of success as, a protein source for a human, for a chicken or a trout or things like that. So that's probably pretty open-ended question, but I'm interested in discussion on that. This really is, what is our measure of success or multiple successes with alfalfa versus another forage crop?
Debbie, do you want to take that one?
Alfalfa has been, really the main focus of research of the forage legumes. Not as much has been done on the clovers, and I assume it was because, the early research really showed that alfalfa has higher production and higher longevity, as well as, producing quite a bit of protein in the herbage. So, success will be, adoption of alfalfa in more rotations so that we could actually see, maybe a return of longer term rotations, including alfalfa with corn, soybeans, wheat, potatoes, other field crops. And, we'd really like to see alfalfa utilized as a feedstock for other products, not just feeding it to animals, and to, raise the demand for alfalfa, as well as increase value to the farmer. Yeah, digestibility is going to vary by what your end product. So when we talk about digestibility, it is for our major consumer right now, and that's the dairy cow. But we are looking, forward to, how can we improve it for fish, for aquaculture, and is that including different oils, different fats? Is it increasing the amount of protein in that concentrate, so you could increase the value to the farmer?
Jeff, does that get at your question?
Yeah, it does. So so it's a similar discussion as the ecosystem services. So it's a matrix in my mind. Is so for the ruminant, here's the positives, but for human consumption, it's probably not the same product. It's not the same alfalfa breeding.
Right.
To do that. So I expect, I'm thinking about it in a matrix, and I assume you guys are, and it's going to be a different answer 5 years from now. You're going to say we have product A, for the ruminant, and very different from product B for human consumption. So that's how I'm thinking about it. So sounds like you guys are similar.
Yeah, from as a breeder, my success threshold or criteria, like 10 years later, if I went to the restaurant, I can order alfalfa burger, and also with omega-3 gene coming out from alfalfa, I will be happy, so I'm successful. Even though maybe my contribution just part of it, but I feel I'm successful for career.
I have a question, Josh. So, well, first of all, I need to address the alpha burger, but I'm from California. We consider alfalfa sprouts to be a staple food product. But so there's a lot of discussion about digestibility and improving digestibility, but I think that runs contrary to the needs of carbon sequestration in the soil, because digestibility probably means low lignin content. And most of the carbon in the soil is lignin. So maybe you need to put these breeders to really a plant that above ground produces very little lignin and sticks it all in the roots.
Right. That's a very good point, Jerry. And there's actually something we're working on looking at right now. I didn't mention it, but we've got a new study going in this spring that I'm collaborating with Jake Youngers and Craig Schaefer on, where we're looking at conventional and reduced lignin alfalfa varieties and impacts on yield and quality, of course, but also total root production, root litter quality, and fine root turnover. And to exactly get at this question, because I had the same hypothesis that if we're impacting the below ground tissue chemistry, that there are some potential implications for carbon mineralization, carbon and nitrogen mineralization too. So the soil mineral dynamics with reduced lignin varieties. Now, whether or not that would make a difference on sort of the longer time, ecosystem level timescales or not is another question. It might involve, some longer term study and modeling, but I think, yes, you're right. That's an important piece.
Jerry will try to convince you to develop an alfalfa tree.
Well, we already have alfalfa trees. Don, we have honey locust and
I know
and mesquite. And
I know.
Illinois bundleflower, right?
China in Hawaii. We already have the alfalfa trees. Don't worry. I just have to get you to fund it through Forever Green.
But yeah, I agree with the points here, Jerry. The breeding goal is digestible biomass eventually. So you have an alfalfa tree, the co-eat, they only digest 1% with 1 ton yield, or we digest 80% with half tons of yield. So probably the digestible biomass my breeding goal instead of the yield. But I follow what your point.
If you go for an alfalfa tree, the only place where they use the vegetative parts would be in Hawaii, where they actually cut down the Lucina and then cows eat it. Most of the places where you grow tree legumes, it's the seed. So it's more like a tree soybean than it is and some of the tree legumes have up to 60% protein. So they're actually better than soybeans in terms of protein content. And so, yeah, that's a whole different area. I bug Julie Grossman on that one a lot, but not others. But that definitely is something, in terms of legumes, that the trees are perennial crop production could be very, very important. And I think you see in Africa, for example, that the acacia are being used to regreen a lot of the desert areas. There's a green band that they're trying to produce across the Sahel to try to de-desertify that area, a critical area of habitat. So there's a lot of good things happening with that. But no, I just thought, since on the one hand, we're talking about digestibility and the other loss of carbon from the soil, it seemed like we were running contrary to two important goals. And it's at least worth considering where that's going to take us. Yeah, you're right. But it's a discussion that Zanyu and I have been having already. So thinking about the other things that we've got the root architecture, the tap-rooted versus fine-rooted varieties. And so thinking about that going forward, not just breeding for the digestibility of the shoots, but what other features of that root system could we be selecting for that are going to help improve carbon sequestration or other environmental services. So
Jim, do you want to get in this conversation?
Yes.
Yeah, thanks I was waiting for opportunity to ask a question. You started talking. First of all, I want to compliment Deb and the team. Fantastic program. I really love to hear about how multifaceted it is. But the conversation about digestibility reminded me like my experience 15 years ago at Monsanto when I was sitting in a meeting. I believe there was a company which maybe the name was Forest Genetics, something with a forest. And they were working on the transgenic alfalfa using RNAi approach. That's why I was in the meeting, where they would downregulate certain phenylpropanoid genes and result in the non-lodging, low lignin alfalfa that is much more digestible. And I do know that Monsanto has developed transgenic Roundup Ready alfalfa. And I do remember it had some sort of issues, but I believe that they were solved. Maybe can you say a few words about where the transgenic, what's on the market, what sort of the large companies are doing in this space, and where is that, was that transgenic low lignin alfalfa commercialized or not? Do you know anything about that?
Yes, so it is marketed. It was released, I think, maybe 5 or 6 years ago now under the trade name of Harv Extra. And so it is an RNAi downregulation of lignin biosynthetic genes. And it's always coupled with the Roundup Ready trait as well. And so they're marketing it as a way that you can delay harvest so that you can accumulate more biomass, but retain quality. A lot of farmers are just harvesting it at a regular schedule and taking higher quality alfalfa. There's still some question about how it feeds to dairy cows. And so Dairy Forage Research Center in Madison is really looking at how this lower lignin alfalfa feeds to dairy cows. So our approach is Joanne Lamb started a breeding program many years ago to select for higher digestibility in the stem. So what we're looking at is a non-GMO type of improved digestibility alfalfa. So
I see. And most alfalfa grown today, at least in the U.S., is Roundup Ready on GMO or not? So is there a market for non-GMO, or it's going to be a niche product? What do you think?
So it depends on where you are in the country. So there are places, I think it's maybe about 30% market share right now. I'd have to look it up, but the latest numbers. But there's, it's not something that is, so a lot of the export countries will not accept GMO alfalfa. So a lot of what's grown for export is non-GMO. It's not very popular on the East Coast, but certain producers really do like it because, they're already using Roundup Ready corn and beans.
Okay, so there's plenty of opportunity to develop non-GMO alfalfa and
Yes.
capture significant part of the market. Great, thank you.
Yep.
Non-GMO will always be important for organics, of course. But the question I wanted to ask Deborah, early on you mentioned something about the nitro alfalfa. And I used it back 30 years ago and would love to have continued it, but it became a perennial crop. And I was just hoping that we could get it to be a crop that would be an annual, especially with the interest in cover crops and in an organic system, a really good potential source of nitrogen.
If you can,
I had heard that in a lot of cases it just didn't die like it was supposed to.
Classic alfalfa.
Yeah. So
Dies when you don't want it to and grows when you want it to die.
Yeah. So we should go back and try to select again for things that will die. So
I would love to work with you on that. Absolutely.
Natural.